Here we are going to begin the new chapter in the medical biochemistry, that is electron transport chain and the oxidative phosphorylation. Before we directly jump into the concepts of ETC and oxidative phosphorylation, let us have an idea regarding energy production as well as metabolism in the cells. Here, let's clear the concept regarding the mitochondrial function inside the cell. We all know that the mitochondria are called as the powerhouses of the cell, and these are the organelles which are responsible for energy production in the form of adenosine triphosphate known as ATP, which is the major form of energy for various metabolic processes inside the cell. So the free fatty acids and carbohydrates are the one which are imported into the mitochondria. For example, when we discuss about the carbohydrates, the pyruvate is the last product of the glucose metabolism, that is glycolysis. So both are imported into the mitochondria, where these fatty acids cross the outer mitochondrial membrane mediated by the carnitine palmitoyl transferase 1 and then crosses the inner mitochondrial membrane with the help of another transporter called palmitoyl transferase 2. So the free fatty acids are metabolized to acetyl coenzyme A by means of beta oxidation in the mitochondrial matrix. And the pyruvate, which is the byproduct of the glycolysis, after entering into the mitochondrial matrix, is decarboxylated to form acetyl coenzyme A by the pyruvate dehydrogenase enzyme complex. The acetyl-CoA then enters the Krebs cycle, which is also known as the citric acid cycle or the tricarboxylic acid cycle, which also occurs in the mitochondrial matrix. So the Krebs cycle is the one which oxidizes the acetyl coenzyme A to carbon dioxide and water, which produces hydrogen ions which reduce nicotinamide adenine dinucleotide called as NAD and flavin adenine dinucleotide called as FAD to form NADH and FADH2. Subsequently, the NADH and FADH2 are used to provide hydrogen ions for the respiratory chain. So the mitochondria are also the site of synthesis of iron-sulfur clusters, which are extremely essential cofactors of respiratory chain complexes, that is complex 1, complex 2 and complex 3, as well as for the enzyme aconitase, which is from the Krebs cycle. These iron-sulfur clusters synthesis requires ATP, GTP, NADH as well as the iron. As mitochondria harbors the electron transport chain, in the electron transport chain, the electrons from the NADH and FADH2 are transferred by various complexes and finally the oxygen. So remember here that once these electrons are transferred to oxygen, then it is converting these electrons by accepting the electrons produces water. So this way all fuels get oxidized to carbon dioxide and water. In other words, in the mitochondria, oxygen is utilized and carbon dioxide generation is supported. Hence, it is called as the respiratory center of the cell. As molecules such as glucose and fatty acids are metabolized, that is oxidized, they produce electrons. These electrons are called as reducing equivalents. So these reducing equivalents, so called as electrons, which are then donated to the coenzymes like NAD plus and FAD to reduce the NADH and FADH2. So these reduced forms of coenzymes needed to be oxidized, and this is achieved when reduced coenzymes transfer their reducing equivalents, which are electrons, to the electron transport chain, and these reducing equivalents are transported within the electron transport chain by means of a specialized set of electron carrier, as well as finally to reach oxygen. That is the reason, remember that oxygen is considered to be the terminal part of electron transport chain and the terminal electron acceptor of the ETC. So this is how in the metabolic processes the reducing equivalents are produced, and NADH as well as FADH2, so produced in the mitochondrial matrix, used for the electron transport chain for the synthesis of an important energy molecule called as ATP, which is also known as adenosine triphosphate.